reproductive system. And because of the way that I've set this up from previous semesters, we did much of the reproductive system stuff in lab. So we're just going to consider the female reproductive system and concentrating on the reproductive cycles and the hormones that regulate that. So the female reproductive system organs are the ovaries, which are equivalent to the testes. That's the gland where egg cells are made. Other than just the functional equivalency that they both make the gametes, they're not that similar. The testis has tubes in it and looks very much like a exocrine gland, which is not exactly true because it's secreting hormones into the bloodstream. But it's got ducts for the sperm to travel through. And then the ovary is much more like what we'd expect for an endocrine gland, sort of just a homogeneous tissue kind of throughout the whole thing. The egg cells, when they're released, are actually rupturing from the ovary, which we'll see in a little while. When they leave the ovary, they go into, let me zoom in on this a little bit. They go into these tubes. Called the uterine tubes, or the oviducts here in the book. They're also referred to as fallopian tubes. They're not actually directly connected to the ovary. When the egg cell is released from the ovary, it has to move into the uterine tube, and these fibri are sort of finger-like extensions on the end of the tube that'll help kind of coax the cell in. It is possible that the cell, when it's released, doesn't get into the uterine tube and can just move into the pelvic cavity somewhere, which isn't usually a problem because the cell will end up being resorbed and probably broken down by white blood cells or something like that and cleared away. However, if sperm get through all of the female reproductive system and get out through the end of the uterine tube and there's an egg cell there and fertilization takes place, that could potentially result in what's called an ectopic pregnancy. Ectopic just means wrong location. And that can be very dangerous here because the pelvic cavity isn't suited for gestation, of course, so it can cause problems. The uterine tube thins down before it attaches to the uterus, and another type of ectopic pregnancy can be if the fertilized egg cell, which fertilization usually takes place towards the end of the oviduct, if that fertilized egg cell, and as it's developing into eventually the blastocyst, if it doesn't leave the uterine tube to get into the uterus, it can implant into the wall of the uterine tube, which is another type of ectopic pregnancy. And that can be troublesome because especially the thinnest part of the uterine tube is, thin, and it can very quickly grow too large in that area and rupture the uterine tube, which would be problematic. So obviously, I've said it a couple of times already, the next part of the system is the uterus, where or between where the uterine tubes attach, the uterus kind of bulges up a little bit. This is called the fundus of the uterus. I just mentioned that because the word fundus we've run across before. There's the fundus of the stomach and the fundus of the eye. So just another place where there's a little bulge in the organ above other structures. We'll look at the structure of the uterus in detail later. It ends in the cervix, which opens up into the vagina, which we see here. The vagina is lined with rugae, just like in the stomach and the bladder, allowing it to stretch out during childbirth to allow the head of the newborn through. This is just another view of those organs. They point to the ovary here. It wouldn't necessarily be here in a sagittal section like this picture suggests, but it would be in this relative location in a more lateral position. The uterus is arranged this direction, so the fundus is more anterior and the cervix is more posterior. And it has a very thick wall. The tissues that make up that wall are pictured in another location, so we'll get to that in a little bit. Now we're going to consider the reproductive cycles that are under hormonal control. There are two cycles that we're going to consider. One's called the ovarian cycle, and that's essentially how the egg cell gets to the point of being released and what the ovary does to support the egg cell after release and up to potential fertilization and implantation. So to begin with, to understand that, let me pull this picture up. This is showing oogenesis, which is the meiotic process of making egg cells, just like spermatogenesis is the same thing for making sperm cells. The stem cell is called an oogonium, which is a diploid cell, and it'll divide through mitosis. One of the resulting cells will remain as an oogonium to give rise to other cells that will go through this process. And the other cell is what's called a primary oocyte. For this particular picture, it's a little strange. They put the label out here because that's where it fits, but it's really what the name of this other cell is. And that's necessary in this picture because they have on the other side here this stoplight icon telling you that cell will start into meiosis. It says meiosis 1, the beginning stage of meiosis. And it actually stops at prophase 1, which is the very, very first stage. And it'll sit there waiting to progress on from when that happens before birth until after puberty. So what this means is all of the cells that are going to eventually become egg cells are initially produced in the fetal ovary. And then they all just sit and wait until there's a signal telling them to progress on further through the process. And those signals won't start coming until puberty. So some of those cells will sit around for wherever in the 10 to 15 years when puberty starts for a girl. And some of them will stick around for the decades worth of time that a woman's reproductive years last. So they just sit there waiting in that first phase of meiosis until they get the signal. And then when the signal comes along, we'll talk about that a little bit more later on, they resume meiosis, completing meiosis 1 and dividing here. Now, meiosis for egg cells, or oogenesis, is different from spermatogenesis because the point is to make really just one egg cell. And that egg cell needs to have lots of cytoplasm to support all of the cellular processes after fertilization. And so the cytoplasm of the primary oocyte is split between the resulting cells. The one that gets all of the cytoplasm is called a secondary oocyte, and the one that doesn't is called a polar body. It says first polar body here because it's the first one that shows up in the process. There's others that we'll see. The secondary, or let me say about the first polar body, it can go on to complete the second round of division in meiosis. It might be that that doesn't necessarily always happen. The polar bodies, as far as anybody understands at this point, don't really do anything. So for the first polar body to divide and make the secondary polar bodies doesn't seem to be crucial, so it doesn't necessarily always happen. I would not be surprised if someday in the future somebody discovers that polar bodies do play an important role in reproductive function, but at this point, nothing's known of that. The secondary oocyte will finish its complete second division. And again, it's going to be asymmetrical, and all of the cytoplasm will go into one cell, and then the other cell is a polar body. The resulting final cell here is the ovum, which is the Latin word for egg. There's a line dividing this portion of meiosis too, just like the one dividing between prior to birth and after puberty. And here, the secondary oocyte will start the second round of meiosis up until it's, or wait until it's penetrated or interacts with sperm. And it's the presence of the sperm that actually stimulates it to complete that second division. So if sperm isn't present, then most of the cells that are released from an ovary are going to just remain as secondary oocytes that never complete that second division. The sperm needs to be present for that second division to take place. So actually there's a point at which the sperm is introducing its DNA as a nucleus, and there's a nucleus of the ovum and a nucleus of the polar body before it gets ejected or before it divides out. So there's actually a lot of DNA here. None of it's mixed together. But eventually the sperm cell and the egg cell will merge their nuclei to make a diploid cell. Yeah.
What does the 2N represent?
So N is the ploidy number. Did I talk about that in lab? So for humans, that's 23. There are 23 pairs of chromosomes. So a diploid cell, which is what most of the cells in your body are, we refer to as 2N because they have 2 times 23 chromosomes in them. And then after the first meiotic division, these cells have just N in them, which is referring to them being haploid, that they just have one of each of the chromosomes. The equivalent picture for spermatogenesis in the book, for some weird reason, has one N instead of N there. It's 1 times something, doesn't change that thing. So 1 N and N both equal 23 for humans. But it's more conventional to just say N for haploid and 2N for diploid. That just represents how many chromosomes are present within the nucleus of that cell. So that's the process the egg cells are generated. Now we want to kind of look at how this progresses within the ovary through a process called folliculogenesis. A follicle is the oocyte surrounded by some supporting cells. Prior to the cells entering into an ovarian cycle, they exist as what's called a primordial follicle. So this is what's present in the ovarian cortex from the time they're generated in the fetal ovary until they're going to be recruited through the hormonal signals. The supporting cells are called granulosa cells. At this point, they don't have the appearance that really is what granulosa is referring to. Granulosa is saying that they're granule or grain-shaped cells, and they don't have that appearance yet. But these are the cells that are going to do that. So at the beginning of the ovarian cycle, a signal comes along and primordial follicles start to develop. The oocyte here, this is the primary oocyte, it's still a primary oocyte here, but those granulosa cells start to develop, to proliferate, divide, and make up a surround encasing the oocyte in the follicle. They continue to divide and make multiple layers of cells. Eventually, and the micrograph down here at the bottom is a better representation of this, eventually space will sort of open up within the granulosa cell region. In this picture, they show space kind of all the way around, which is misleading. But when we see either small spaces start to develop or one large space form within that, that's what we call the secondary follicle. And what's misleading about this picture here is that the oocyte is still attached to the edge of the follicle through a mass of granulosa cells. It's not evident in this, and it might just be that the picture that this is drawn from would just not cut through the follicle in such a way to see that area that it's attached. But with this one, we can see that the egg cell is not ever completely removed from the outside of the follicle. And then as the follicle grows larger and larger, it becomes the tertiary follicle. And tertiary just means third in order after primary and secondary. I prefer to call it the mature follicle, just because tertiary is not a very common word. But it's the last stage of follicular development. And here we can see the oocyte is definitely anchored to one side of the follicle. The empty space represented here in the secondary follicle is present as what's labeled as the antrum here. And in the micrograph, we label this as a developing antrum. The antrum has cellular debris from the granulosa cells breaking apart and forming that space, as well as fluid, which is what's kind of represented by this gray structure that seems to be labeled as the antrum. I'm going to mention these later, so let me point them out. In the secondary and tertiary follicle pictures, there are cells on the outside edge of the follicle. They're not actually part of the follicle, called theca cells. And I just want to mention them here because they're going to be relevant later on. The reason I like to call this the mature follicle is because it's the last stage of the follicular development, like I said, and what's going to happen next is it's going to rupture and release the oocyte. The oocyte that's released at ovulation is still surrounded by some of the granulosa cells, but the majority of the granulosa cells stay behind as the wall of the ruptured follicle. And then the oocyte will go into the uterine tube, as I was talking about earlier. What is left over of the follicle in the ovary becomes what's called the corpus luteum. So the rupture closes back up, and the granulosa cells that were there start to change and make up what's called the corpus luteum. The name corpus luteum is referring to the fact that it has a yellowish color to it. Luteum refers to it being yellow in color. It's going to persist for the rest of the ovarian cycle and release hormones to help support the egg cell if it becomes fertilized and implants into the uterine wall. But if that doesn't happen, then the corpus luteum will degenerate down to what's essentially scar tissue, at which point it's called the corpus albicans, and we'll see that word later on. Albicans, related to the word albino, is referring to it being white in color at that point, which is basically scar tissue. So the first picture here, the primordial follicles, prior to an ovarian cycle. So the primary, secondary, tertiary, or mature follicle pictures, ovulation, and the corpus luteum picture represent the ovarian cycle. The ovarian cycle is on average 28 days long. And the midpoint is ovulation. Day 14 is ovulation. So the 5 pictures we're looking at here, 3 of them represent the first 14 days, and 1 of them represents the second 14 days, and then 1 of them is the midpoint between the 2. So in these pictures, it doesn't look like it's quite balanced for the first half and second half, but that is in fact the case. The first half of the ovarian cycle is referred to as the follicular phase because the follicles are, for the sake of recording, let me do this here. Follicular phase referring to the fact that it's when the follicles are developing. And then after ovulation, the second half is referred to as the luteal phase, referring to the presence of the corpus luteum. Now, actually, let me say this a little bit differently. Ovulation does not take a whole day. So let me just say that days 1 through 14 of the follicular phase, on day 14, ovulation occurs, and then days 14 through 18, after ovulation, is the luteal phase. So I just want to introduce those terms. They'll come up in a picture that we'll look at a little bit later on. But this is the book's representation of what the structures look like as they're doing this. Hormones control the process here. During the follicular phase, what's guiding it is the hypothalamus releasing gonadotropin releasing hormone, which I mentioned in regards to the male reproductive function. And gonadotropin releasing hormone causes the anterior pituitary to release the gonadotropins. And the gonadotropins are FSH and LH. We met those before. Again, they regulate male function as well. Here, I'm going to tell you what they stand for because their names are actually referring to the female function. FSH is follicle stimulating hormone. So when it's released, it stimulates the primordial follicle to start to develop into the primary, secondary, mature follicles. I'll hold off on LH for a second because what it does isn't relevant until the luteal phase. When the follicles are stimulated by FSH and LH technically, they start to develop and they start to release a hormone called estradiol, which is the primary estrogen hormone. That will then go ahead and tell the uterus what to do, and we'll talk about the uterine cycle in a little while. But also estradiol works as a negative feedback signal, telling the hypothalamus and the anterior pituitary to stop releasing those hormones, GnRH and FSH and LH. Then at probably more around day 12 or so, the hormone changes leading to ovulation are based on the fact that estradiol levels will get very high. So the thickness of the arrow in this picture represents just a higher concentration. And when estradiol gets to its peak concentration, that actually switches it from a negative feedback signal to an activator, which then tells the hypothalamus to release all the GnRH it hasn't been releasing. As a negative feedback signal, estradiol doesn't tell it to stop making GnRH. It still makes a GnRH, it just doesn't release it. And it also doesn't tell the anterior pituitary to stop making FSH and LH, but rather tells it to stop releasing it. And so the burst of GnRH that comes out from the hypothalamus will tell the anterior pituitary to release all of the hormone that it's been saving up. The thickness of the arrows for LH and FSH is relevant here also. The LH arrow is a little bit thicker than the FSH arrow because at ovulation, more LH it gets released than FSH. The peak peak of LH release is significantly more than the peak of FSH release. And that causes the follicle to rupture and release the oocyte for ovulation. Estradiol is still having an effect on the uterus. Then the third part of this figure is showing what happens in the post-ovulatory part, which is called the luteal phase. Now, LH stands for luteinizing hormone because it causes the luteal phase to take place. It causes the corpus luteum to form. It actually does that by causing ovulation. Once ovulation takes place, then the remainder of the follicle becomes corpus luteum, and the ovarian cycle goes into the luteal phase. So FSH and LH really don't have much effect anymore at this point. But because of what happened in ovulation, luteinizing hormone, which is the primary thing that causes ovulation, has caused the corpus luteum to form. The corpus luteum actually still does release a little bit of estradiol, but now it starts to release another hormone called progesterone. Progesterone takes over the negative feedback signal that estradiol had prior to ovulation, limiting the amount of release of GnRH and FSH and LH, and also stopping production of it. It's not being saved up by those glands anymore. It just stops being released for the rest of the cycle. Progesterone also starts to tell the uterus to do something a little bit different. We'll see that when we look at the uterine cycle in a little bit. So these are the representations of hormonal control. During the follicular phase, GnRH tells the anterior pituitary to release FSH and LH, which stimulate follicle development. The follicles release estradiol, which will influence the uterus, but also act as negative feedback signals to limit the release of these hormones from the hypothalamus and anterior pituitary. When estradiol levels get high enough at their peak, they'll then switch to stimulating the hypothalamus to release the GnRH that it's been storing up, which will cause the anterior pituitary to release the FSH and LH that it's been storing up. LH becomes the major signal here and causes ovulation, which then results in the corpus luteum forming from the remnant of the follicle. The corpus luteum starts to release progesterone now, which has slightly different effects on the uterus, but still acts as that negative feedback signal to the hypothalamus and anterior pituitary. So, this image is showing both what the ovary looks like as a tissue. All of what happens in the ovary that we've been talking about is happening in what's called the ovarian cortex. If we have a cortex, there's also a medulla in the ovary. The medulla is really just where blood vessels and lymphatic vessels, that sort of thing, enter into the ovary. More importantly, this picture shows us something about the structure of the uterus. The uterus has 3 layers of tissue making up its wall. The innermost layer is called the endometrium. The thick muscular layer is called the myometrium, and then the outer covering is called the perimetrium. What's not indicated in this picture, but is true, the endometrium actually has 2 layers within it. I prefer to call them the functional layer and the basal layer. Just that's the English equivalents of other names that are given to them, which are Latin. Those are just functionalis and basalis, but it's pretty obvious which layers that refers to. The basal layer doesn't change a lot. It's just connective tissue that anchors the endometrium to the myometrium. The functional layer has the endometrial epithelium, which is a simple columnar epithelium, and connective tissue that changes in size throughout the uterine cycle. Yes?
What was that? Endometrium?
Yeah, the functional layer of the endometrium grows throughout the uterine cycle. This figure actually shows a lot of different things going on. For the sake of what we're talking about here, let me focus first on the uterine cycle. Zoom in on it. Come on. It pretty much fits there side to side, but this is based on the 20-day cycle, and it's the same 28-day cycle as the ovarian cycle. The uterine cycle is separated essentially into 2 phases, pre-ovulatory and post-ovulatory. Pre-ovulatory is called the proliferative phase. Post-ovulatory is called the secretory phase. There's a reason why the section that's called proliferative phase seems to be only half of the first part, but that's what we call the pre-ovulatory part. The reason for that is actually the beginning of the uterine cycle from day 0, which is day 0 for the ovarian cycle, actually shows the tissue of the functional layer sloughing off in what's called menstruation. It has a rather significant blood supply to it, and so the menstrual flow is bloody from that. Once that sloughs off after about 5 days, then the endometrial wall, the functional layer, is going to grow back. It's going to proliferate. The cells that make it up are going to proliferate and they'll grow thicker. So that's why it's called the proliferative phase. After ovulation, it's still basically growing, but now, as the corpus luteum becomes active in the ovary and releases progesterone, progesterone causes the secretory phase. These big lines going down in through the endometrium are endometrial or uterine glands, and they secrete mucus, which helps to protect the inside of the uterus and also make it more hospitable to the potential implantation of the blastocyst. It'll contain nutrient molecules to help the blastocyst survive in the endometrial space until a connection's built up that's going to eventually become the placenta. And some of what's secreted there can also help power sperm cells that are swimming through here. This ejaculate contains fructose to help power ATP production of the sperm. But by the time it gets to the uterus, the ejaculate's really diffused away so much that there's not going to be a whole lot available for it to use for energy. So it can use what's secreted in the uterine cycle for power. Glucose or fructose or whatever's secreted there. At the end of the 28-day cycle, the functional layer will then start to slough off, leading into menstruation, which continues on at the beginning of the next cycle. We'll talk about in a little bit what's the signal that causes that to happen, but it finishes out there. Now, I want to take a second. Hold on a second. I don't want to search for that. Putting sex into a Google search is never a good idea when you're recording a lecture. All right, so. This might be good. Okay, so there's a lot going on in this picture. I just want to kind of give you an impression of the relationship between the hormones we're talking about. So testosterone and estradiol and progesterone are all steroid hormones. And being steroid hormones, that means that they are derived from cholesterol. Cholesterol has this typical ring structure here that we see throughout all of the steroid hormones. And really the differences between the hormones are based on what's attached to the ring structure on the outside. So cholesterol has this carbon chain structure on its outside, which is called the cholesterol side chain. And there's an enzyme that basically cuts that off and leads us into the series of what are going to be steroid hormones. What's made after that part of cholesterol is cut off is then modified by one enzyme, and that's progesterone. And then there are a number of other possible places. Progesterone is basically the precursor for a lot of the mineralocorticoids like aldosterone and cortisol, which have come up in this course before. But also there are enzymes that will modify that. When we get down here, we're starting to talk about the sex hormones. So this hormone here, which I'm not naming on purpose because I don't need you to know the name of it, is the precursor to testosterone and to a hormone called estrone, which is another estrogen hormone. The enzyme that converts this into estrone or converts testosterone into estradiol is called aromatase. Aromatase is an enzyme that creates an aromatic ring in one of these molecules. Now let me see. Yeah. So this is a 6-sided carbon ring. And it's referred to as aromatic when it has 3 double bonds in it. So for sort of the pictures we were just looking at, this is what a ring would look like. But if we drew in all the carbons and hydrogens, this is what it would look like. That's an aromatic ring. Okay. So if you look at estrone or estradiol, they both have an aromatic ring in this position because they were both made by aromatase. If we look back at testosterone in that position, the only double bond in that ring is right here. And so what aromatase does is it takes this bond, this ring, and changes it. Now, don't get too bogged down with the chemical structure I'm talking about here. I'm sort of leading up to making a point. So this is that end of testosterone. It's got a double bond here. It's got a double bond to an oxygen out here. There's a carbon sticking up here, and then this is the beginning of the next ring over here. What aromatase does is it breaks a double bond to oxygen, so a hydrogen attaches to the oxygen. Rearranges the double bond here to put it over here, puts a double bond in there, and puts a double bond in there. This double bond now makes it impossible for this carbon to be attached to something here, so that's removed. And that's the end of estradiol or estrone. What's happened here is a hydrogen has been added And then where this double bond's put in, 2 hydrogens are removed, if I remember correctly. I think, no, this one stays the same. This one stays the same. And then here, a carbon is removed and 3 more hydrogens are removed. So really the difference between estradiol and testosterone is the net change that there's 5 new atoms attached. All right, 5 atoms removed, sorry. Okay, so it's a very, very small change in the overall molecule. Let's say, okay, so there's 18 carbons in estrogens, 19 in androgens, which include testosterone. And that's not counting the hydrogens that are there, and there's always an oxygen or 2. So 5 atoms is a really, really small variation between these 2 molecules. But testosterone and estradiol have profoundly different effects. What makes testosterone act so much differently for human physiology than estradiol, or vice versa? What's the difference between those 2 hormones if, chemically speaking, they're only different by a few atoms? What makes hormones behave differently? How do they have their effects?
The effects of different enzymes on them.
Hormones don't interact with enzymes. What do they interact with?
Organs.
Well, they're chemicals. They have to interact with something at the chemical level. How do they interact? I mean, yes, they interact with organs, you could say, but it's because they interact with cells. But they interact with cells because they interact with something else at the chemical level. What do hormones actually interact with?
Blood.
Well, blood's a tissue. Something at the chemical level.
Cell membrane.
That would still be at the cellular level. What in the cell membrane do hormones interact with?
Receptors.
Receptors. Now, what you said actually was going down the wrong path. These are steroid hormones. They're lipids, so they just pass right through the cell membrane because they diffuse through it. But it got me to where I wanted to get, which is receptors. So testosterone and estrogens have different receptors. So when testosterone binds to its receptor, or estradiol binds to its receptor, they become very, very different structures. And so they have very, very different effects, even though the hormone itself is fairly simple. These 5 atoms that are changed, or 6, however you want to say it, is enough to mean that testosterone can bind to one receptor and estradiol binds to another receptor. Okay. And that has very profound effects on cells and tissues and organs later on. Okay. So I just want to take this little side trip to kind of point out the structure of these molecules and how they're related, but also how they fit into the bigger group, which includes other steroid hormones that we've come across. And they're all related family-wise. And progesterone, which plays a part in reproductive function, is a kind of general steroid hormone that's a precursor to a number of other things, but it's used itself to have effects that we're about to talk about. Not there. Unzoom. Oh, shoot, I didn't want to unzoom. That would be here. So, if I had made this picture, one thing that I think is really needs to be done is the 0 to 28 day scale on the 2 pictures here needs to be the same way, because day 0 in the uterine cycle is day 0 in the ovarian cycle. Day 14, day 14, day 28. They're the same scale, but for whatever reason, The uterine cycle picture was drawn slightly different. What happens at the beginning of the ovarian cycle is actually drawn up above because it doesn't really fit in the space they have here. But we already talked about this. FSH will stimulate primordial follicles to start developing into primary and secondary follicles and eventually into the mature follicles at the end. The point that this is making is that multiple follicles will go through the developmental process, and only one of them will eventually be selected to be the one that's going to be fully mature and rupture at ovulation. So just where the arrow is, that's all the stuff leading up to the maturation of the follicle. And then at day 14, it ruptures. So everything up to that point is the follicular phase. And then after ovulation, we have the luteal phase, where the remnants of the follicle become the corpus luteum. In this picture, they show sort of a suggested straight decline down in size. This is probably a little misleading. I'd expect the corpus luteum stays sort of full size longer, and then at the end degenerates down, depending on the hormonal signals that will regulate that. But so that's the ovarian cycle that we talked about earlier, and we can see how it lines up with the uterine cycle. Also, we want to consider how hormones play a role here. So these two graphs at the bottom show the concentration of the pituitary hormones in the blood and the concentration of the ovarian hormones in the blood. Again, set up on the same 28-day cycle. So at the beginning, because of the release of GnRH, which is not in this graph, FSH and LH levels start to grow. FSH is predominant between the two, not a huge difference, but it is predominant because it's stimulating the follicles to develop. The levels don't get very high, and they dip down here because estradiol levels are increasing. Now in this, they label this estrogen, but it's specifically estradiol that's being released here. And so as estradiol levels increase, that suppresses the release of FSH and LH, and their levels go low. But then when estradiol gets up to its peak levels, that's going to stimulate GnRH to stimulate the release of FSH and LH. And the LH levels will be huge. The FSH levels go up a little bit too, but they're a minor component. And this is what causes ovulation. And then after ovulation, LH and FSH levels drop back down because they're done doing what they need to do. But after ovulation, the corpus luteum forms and it starts to secrete progesterone. Estrogen levels have gone down, but they come back a little bit at this point too. So post-ovulatory, during the luteal phase of the ovarian cycle, progesterone levels go up and estradiol levels are kind of coming up too. And that's what stimulates the secretory phase for the uterus. What causes the endometrium of the uterus to fall off is actually the drop off in mainly progesterone, but progesterone and estradiol together is what leads to menstruation occurring. Now, birth control pills are based on the ovarian hormone levels that we see here. Your standard birth control pill regimen is going to contain progesterone levels, and especially some of the more modern compositions, low estradiol levels in it. But at levels to mimic the amount of those hormones that are released from the ovaries post-ovulation. In your standard 28-day cycle of birth control pills, there's a series of 7 pills that don't contain any hormones to mimic that drop-off in progesterone levels, and that leads to menstruation. but then immediately the next pill that's taken after that last non-hormone pill, the levels go back up to the post-ovulatory level, which tricks the body into thinking that ovulation's already taken place, and so follicles don't develop and ovulation doesn't take place. So that's how it's an effective birth control measure. There's nothing that necessarily says that has to follow a 28-day cycle. And so there are other regimens for administering hormonal control for birth control. There's a brand of birth control pills called Seasonique, which instead of working on a 28-day cycle, work on a 3-month cycle. The name Seasonique referring to you take a dosage for a season, and then there's a week-long no hormone pill sequence that would stimulate menstruation, and then it happens again. If you watch the Seasonique commercials, I haven't seen any much lately. Of course, I don't watch as much television as I used to because I got a kid now, right? But it used to be fairly common. And there was fine print in the commercials that would say, does not necessarily reduce the occurrence of monthly spotting or monthly bleeding or something like that. Because there are probably other signals to the endometrium to initiate sloughing off, and so it's not uncommon for that to happen. But as far as the hormone levels are concerned, there's nothing that says that they have to fall off after 28 days. They can persist. And in fact, if fertilization implantation takes place, the corpus luteum will remain in the ovary for the next couple or few months, releasing progesterone until the uterus takes over all of that function itself somewhere towards the end of the first trimester of pregnancy. So having hormones released for a long time is what happens when pregnancy's taking place, and so it's going to just suppress ovulation even further. There are other ways of administering hormones. There's a patch. There are hormone-infused IUDs. I don't know how those regimens work, but they're still basically doing the same thing of mimicking the post-ovulatory hormone levels. IUDs also are physical presences in the uterus, and so they also help to stimulate the uterus into sending out signals saying that something's implanted into the surface, into the endometrium, which is just further going to suppress ovulation. So this figure is just getting at putting the ovarian and the uterine cycles together and looking at the hormones that tie all of that together. I want to remind you of this picture that I showed you. Where is it? In talking about the male system, where you first heard about GnRH and FSH&LH, which has an effect on cells within the testes that stimulate spermatogenesis. It's the exact same hormones that are at play for the female system. There's no reason to have a whole new set of hormones for the male system. They're just adapted to what's there and carry out the same functions or analogous functions in the male system, but they are related to each other. So. Any questions here? Everybody's good with this? Okay. So I sort of designed the end of the semester of this course somewhat hastily, maybe, in semesters where I got kind of behind with things. And so I did the introduction and male reproductive stuff in lab and female reproductive stuff in lecture, just to deal with the possibility of running out of time. And there have been many semesters in the past where I had only one day to cover the reproductive system lecture, and so I sort of tailored this to that. That's all I have to say about the reproductive system. I mean, between lecture and lab, we've covered it quite a bit, but I'm not going to go any further. I suppose there are other things to do. There's another chapter on fertilization and development. I pulled a couple of pictures out of that in the stuff I showed you in lab about division and chromosome and that kind of stuff, cell division and chromosomes and that kind of stuff. But I've covered all the things that I want to cover for the reproductive system here. But of course, we're not as behind this semester as I have been other semesters. So I'm done. We do not have to have class on Tuesday next week. Okay. Monday is an optional dissection day, so if you didn't sign up for the dissection, you don't have lab either. So you have plenty of time to focus on getting stuff done on Blackboard before the end of the semester. Which is next Thursday. Okay, so remember we have our final exam period, 11 o'clock on Thursday the 12th. We'll be in room 500, the lab room, to take the in-class portion of the second practical. And when you come in, I'm going to shut off access to Blackboard. So you need to have everything done on Blackboard that is there, so that nothing untoward comes of people saying, oh, I need to go finish more stuff. You don't get any chance after that. So if you don't have any other questions, that's it.
I just got a question. Final exam practical?
So the final exam for the course is actually an online assignment. So we use the final exam period for you to take the second practical.
Okay. Yeah.